All of these are going to be available on the YouTube page as well, yes?
Yep. So
Yeah.
after we finish, yeah, exactly. So I just started recording, and as soon as this is over and like our review session done and everything, it'll take some time to upload, but it'll be up there.
Awesome, thank you.
No problem. So yeah, so this is week 3 topics. All right.
Okay, so let's begin. And the first topic we're going to talk about this week is protein trafficking, which is a good chunk of what you're going to be learning this week anyway. So we're going to start initially with how are proteins delivered. And this is going to be for eukaryotic cells only. So just keep that in mind, like prokaryotes will use a different system. And so basically the idea is proteins are location specific, right? So certain proteins are localized to the nucleus and other proteins are localized to different areas like the ER or whatnot. And so for those proteins to function properly, they have to be in the proper place in the cell. And so protein trafficking is the way the cell manages to move the proteins to their specific location, right? And in eukaryotes, they use a signaling system involving amino acids. So you can think of it like a postal service essentially, right? So, right, when you try to mail a letter, you write the address you're trying to send the mail to. The mailman reads whatever address is on there and they interpret it and then they send it to the correct site. And so that's basically what the cell is doing, right? There's going to be a tag. We're going to talk about those later. There are like tags on proteins. They're interpreted by other proteins in the cell, and those proteins act as mailmen delivering proteins around the cell. So that's like the overall concept. Yeah. And so we really have to begin with protein synthesis, right? So the beginning of any protein is really translation through a ribosome in the cytosol. And at this point, there is a branch. The protein will either go through the endomembrane system or they will not. And so we're going to go over the endomembrane system a little bit more. Next slide. Thank you. So what is the endomembrane system? Essentially, is a system of membrane-bound organelles that work together to modify, package, and transport different proteins and lipids. And right, so we see the free ribosome. It starts the translation, but during translation, the ribosome will actually travel to the rough ER, and the protein will be transferred once it's fully translated into the rough ER. And from there, you can see it will travel to the Golgi body through vesicles and either to other places like the lysosome or the plasma membrane to be secreted. So one thing to notice about the endomembrane system is really like it shares the lipids. So lipids in the ER, they'll bud off to form these vesicles, which are just like little transport vehicles for proteins. And so those budding, essentially like mini membrane systems, will travel to the Golgi body and they'll be incorporated into the Golgi body. And then the Golgi body has its own budding system, right? And it'll either go into the lysosome. So the lysosome is another organelle that buds off the Golgi body and it has functions in like catalytic functions. Or it can go into the plasma membrane. And when those secretory vesicles fuse with the plasma membrane, their insides will be secreted outside of the cell. So the endomembrane system is really just a way for the cell to create proteins, modify them, and help them be secreted later. So a couple important notes to think about. Endomembrane proteins remain in the endomembrane system. Once you enter the rough ER, you will not leave into the cytosol anymore. Once you enter the rough ER, you have to travel along that system, right? So rough ER, vesicle, Golgi body, lysosome, or like the plasma membrane. The only exception to this would be the unfolded protein response. But that's just one exception. Generally, they stay within the endomembrane system. And then we can move on. So here would be a visualization. So we see the cytosol the protein would move to endoplasmic reticulum, into the Golgi body, and then into different sectors. Here's just more visualization. I think there's another one after this. You can go off, you can look at these pictures in your own time if you have any questions. Yeah, next. And so, right, so that's the endomembrane system. Proteins, some proteins aren't part of the endomembrane system. And so essentially, if you're not going to the rough ER, the free ribosome will translate that protein in the cytosol, and that protein will either travel to the mitochondria, the nucleus, or it will remain in the cytosol. It could also travel to other organelles like the chloroplast, but we won't really talk about that in this class. And so, right, if we're thinking about this, how do cells actually identify proteins? And they do it through signal sequences. And signal sequences are essentially amino acid sequences that have specific properties, right? So when we remember, like, primary structure develops into secondary and tertiary structure. So these stretches of amino acids will create a fold in the final protein, and it will have a specific property that carrier proteins will recognize and bind to. And that's really all a signal sequence is. It's a binding site for other proteins to interact with. And so there's different types of signal sequence we'll talk about in this class, but essentially you can break them down into two types. There's localization and retention signals. And so the endomembrane system really uses the ER localization and different retention signals, but there's also other localization sequences like the nuclear localization sequence and the mitochondrial localization sequence. We'll just go over that in the next couple of slides. And right, so here's an example. If we look at the ER localization sequence, you'll see there's just stretches of hydrophobic amino acids near the N-terminus. While if you look at the mitochondrial localization sequence, it has interspersed positively charged amino acids. And so essentially, right, like it's just, they have different properties that different carrier proteins can recognize. And so the endomembrane signal sequences, right, we went over this just briefly, ER localization and the retention sequence. So the most important signal is the ER localization sequence, because that is what tells the cell that this protein is destined for the endomembrane system. And so, right, the initial entry point into the endomembrane system is the ER. If you don't enter the ER, you won't enter the endomembrane system. And so what happens is, right, co-translationally, while translation is occurring at the ribosome, and remember, the signal sequence is near the N-terminus. That's one of the first parts of the protein that is translated. So it's immediately recognized during translation. A protein called SRP, or the signal recognition particle, will bind the ER localization sequence, and it acts as a carrier protein, bring the ribosome complex towards the rough ER. And this is essentially the entry point. And then if you look down at the graphic, right, the analogy would be like if you're taking a bus, the ER localization is your bus ticket. It's how you get onto the bus. And then along the way, there are retention signals. And if the cell detects a retention signal, it'll just keep the protein in that specific place. So there's different types. There are like ER retention sequences, Golgi retention sequences. So those retention sequences tell the cell that this protein is meant to stay in the ER. This protein is meant to stay in the Golgi body. So that would be like an early exit if you grab like the wire on buses. And if you have no retention sequences, right, you just travel along the endomembrane system, and you'll eventually just be excreted. That's like the last stop, and you have to get off at that point.
I just wanted to say I really like that analogy. I haven't heard that before.
Thank you. And so, right, fundamentally localization sequences, like we just said, their localization is when your protein travels through a membrane. So the mitochondria, right, mitochondria is membrane bound, or the ER is membrane bound, and so is the nucleus. The proteins can't just freely diffuse into these organelles, right? Like it's regulated through channel proteins. And so the localization is fundamentally, right, the cell recognizing like this protein needs to travel through this specific channel into an organelle. So the mitochondrial localization sequence would just be recognition of a mitochondrial transmembrane channel protein for that target protein to enter the mitochondria. And so is the ER. Like the ER and mitochondrial localizations are very similar. The nuclear localization is a little bit different because the nucleus uses nuclear pores, and those aren't exactly the same as channel proteins. We'll get into that in the next slide. So what is the mechanism of localization? Essentially, in the ER and the mitochondria, the channel itself in the channel proteins are too thin to allow a fully folded protein to enter. So, right, when the channel opens, the unfolded form of the protein will enter into the mitochondria or the ER, and inside the organelle, chaperones will help the protein refold. In nuclear proteins, it's a little bit different because the nuclear pores are much bigger than a channel protein. Nuclear proteins can enter the nucleus fully folded. And another difference between ER, mitochondrial localization, and nuclear localization is that after a protein enters the ER with a mitochondria, their localization sequences are cleaved because those proteins will essentially never leave the cell again. Or not the cell, it'll never leave those organelles ever again. But the nuclear localization sequences can't be cleaved because nuclear import and export is a little bit different. We'll get into that later as well. And so if we look at this diagram, right, this is like a flow chart. You start with a free ribosome. And as you start translating, the localization sequence will appear. If it's an ER localization, it'll be taken co-translationally to the ER. And at that point, it enters the endomembrane system. If it has a retention sequence, it stays within the endomembrane system, somewhere in there. If it doesn't have a retention sequence, it will be secreted. And if it doesn't have the ER sequence, it'll have a mitochondrial or nuclear localization, and those target for those respective organelles. And essentially, if you have no localization sequence at all, right, you'll just stay in the cytosol because there's no carrier proteins to traffic you anyway. You're just sit in the cytosol. And here's more diagrams. We can just go next. So here's just a quick summary of everything we just said. You can read it in your own time if you'd wish, but we'll probably just go through this right now. So let's just do a quick practice question.
So a model of the types of questions oh my gosh, I can't believe I just said that. But these practice questions are a model of the types of questions that Laura would likely ask on an exam or problem set. So just to get you more practice with these type of questions.
So a transmembrane proteins must have ER import signal sequences on their N-terminus. Predict where you would find a membrane receptor protein if you engineered its genes such that you remove the ER signal sequence. Assuming that your receptor protein does not completely misfold or get degraded, which is always a possibility when you manipulate genes, where would your receptor end up? And so, right, if we think about this situation, the first question we should ask ourselves is, what localization sequence does it have? Because that will determine its initial state. Does anyone have an answer to that?
And if anyone does have an answer, feel free to either unmute yourself or you can type it in the chat if you want.
Okay, well, if no one's gonna say anything, we can just answer
Yes.
it ourselves, I guess. So essentially, right, it doesn't have a localization sequence because you removed it. And without any localization sequence, it's not going to be imported into the endomembrane system. It's not going to be imported into the mitochondria. It's not going to be imported into the nucleus. So the only other option really for it is just to sit in the cytosol somewhere, essentially. We could ask like a modified question. What if in your protein, right, you have the protein naturally has a nuclear localization sequence. What if you mutated it to also add an ER localization sequence? What would happen to the protein?
I think this was one of the clicker questions from class today. So if you were in class, you would know.
Come on, guys. Is no one paying attention to the lectures? Would
Oh,
it
wait,
go
we
to
have a
the
chat.
ER?
We have a chat.
Okay, okay.
Would it go
Angela
here?
says, would it go to the ER? And yeah, that's essentially the answer, right?
Yeah.
And when you're answering that question, you're trying to look at, it's a competition between which localization sequence gets recognized first. Does the ER recognition occur first, or does the nuclear recognition occur first? And right, if we think back, ER localization occurs co-translationally, while the nuclear localization occurs after the protein is fully folded, because the nuclear localization sequence is located somewhere on the outer fold of the protein. It's part of its three-dimensional shape, right? And so if the ER localization is recognized first, the protein will be inducted into the rough ER. And as we said before, once you're in the endomembrane system, you're basically in there forever. You're either in there somewhere or you get secreted. And yeah.
Okay, and just to address like Elena, that's totally fine if you weren't in class today. Might think about like being in class, that's fine. But if you haven't started like Moodle or anything like this week's content wise yet, that's totally fine. It's good to even just hear all this information more than once. I forgot to say in the beginning, but Tuesdays, our review sessions are more like content based. And even if you haven't gotten to all of the week's content yet, we offer like this review session to kind of like put it out there for students that come so that you have like kind of a basis of it. And then once you learn it through Laura's lecture videos, then you can have like a more in-depth understanding. And then by Thursday, that review session, you can really go in with the practice questions that they have and really get into like how to answer questions and use the knowledge that you've learned from either today and all the other lecture videos into like exam like or problem set questions. Okay, cool. Oh, where did my mouse go? I've lost my mouse. Oh, there it is. Okay. All right. This was me, right, Robert? Sorry, I forgot.
Yeah, I'll hand it off to you. I'll just shut up.
All right, sounds good. You did it lovely. Okay. So secretory pathways, these are just some examples of when a product or something inside the cell cannot or needs to go out of the cell. And these are like real life examples of ways that secretion works within a cell. So for this one, this was also an example, I believe, in class today, but it's hormosecretion. So essentially what's happening here is you have a mRNA, this hormone, being bound to the surface of an ER. And then once it goes into the ER, so then that indicates to you that it must have had some sort of ER localization sequence. And once it's inside the ER, that signal sequence gets cleaved off, as you see over here. And now it's just known as a prohormone in this scenario. Because this did not have a ER retention sequence, it's going to continue on through the endomembrane system. So it's going to go out of the ER through the Golgi. It's getting chopped up into pieces along the way. And eventually these secretory vesicles are going to transport it out of the cell through exocytosis. And then that's just the word for when, like, if you want to think about it, like a cell basically like throws up whatever is inside that it doesn't want. So that's the opposite of endocytosis, which is when a cell wants to bring in something. And then this is just another example of a real life protein sorting, protein trafficking. So these lysosomes are involved in waste removal, as a lot of people know, but specifically how they work is that a lysosomal enzyme is formed and it goes through the ER it goes through the ER with its localization sequence, but it doesn't have a retention sequence. So it's going to continue on through the Golgi and travels essentially through the entire endomembrane system and gets vesicled out of the cell or to wherever it needs to go as well. And then Robert, I think there's like some things in the chat if you want to handle that.
Yeah, I got it.
Thank you. Okay, so Robert had said before briefly about nuclear import and export and how that's a little bit different. A lot of students get confused about like the whole cleaving off of signal sequences in nuclear import, export. That doesn't really happen because, again, their process in itself is a little different. I believe these are one of the lecture videos that are like later on in the week technically, or one of like the later lecture videos. But again, like I said before, it's just a nice little review baseline. So if you don't completely understand right now, that's okay. Feel free to ask questions, or it'll help give you a more better foundation for when you do eventually watch Laura's lecture videos. Okay, so kind of just a umbrella term. Nuclear import and export is regulated by the GTPA's RAN. And GTPAs is like this big super family. It has a lot of different enzymes and proteins located under it. So it's like this huge umbrella term, as you can see from all these, from all of these different pictures. RAN is just one part or one member of this family. As you can see here, its family tree is a lot smaller than all the other ones, but RAN is a member of the GTPases, and one of RAN's siblings, so to say, would be RAS. And then these are really important in like kinase cascades and just like a little connection to bio 151 in case you want to make that connection. Sometimes it's really helpful for students to make connections between their different classes, but we will focus on RAN. Okay, so in order to understand nuclear export and import as well, GEFs and GAPs are really important to understand and what they do. If you don't really know what GEFs and GAPs do, it's hard to visualize what's actually occurring within the nuclear import-export cycles. So GEFs and GAPs are proteins that regulate the RAS family of GTPases. We'll start with one at a time. So we'll start with GEF. GEF stands for guanine nucleotide exchange factor. So you can think of it as it's exchanging the GDP to GTP. So the diphosphate to a triphosphate, and the GTP state is when the entire GTPase is active. So it's going to be doing its job in the nucleus. And the end result is that RAN GTPase is bound to GTP. So it used to be bound to GDP, but then GEF comes in to help it out, and the GDP filters out and GTP naturally filters in. So that's one of the most common misconceptions. Students think that like GEF changes something to make GDP turn into GTP, but rather what happens is it's a conformational shift. So if GTPase was first bound to GDP, then once GEF comes in, it causes the affinity, which we learned last week. So the affinity between GDP and the GTPAs to decrease. So it's going to let go of that GDP. And then naturally in the cell, there's a lot of GTPs floating around inside the cell. So it's in high concentration, and GTP can easily bind into the GTPAs. Okay. I know that's a lot of like GTP, GDPs. It's a mouthful to say as well, but there's no really better way to put it. So if you have any questions, feel free to stop me at any point. Okay, so then there's GAP, which is the opposite of GEF, and that stands for GTPase activating protein. Essentially what it does is it hydrolyzes GTP into GDP. So that is the end result. Instead of it being bound and active, it's now inactive with GDP. One fun way that a student had mentioned to me, and it's really easy for me to remember now, is that a gap, you're literally like forming a gap. So instead of GTP, which is 3 phosphates, you're cutting it so that there's a gap between the phosphates, and now you only have 2 phosphates. So if you can remember that gap causes it to become bound to GDP, then you can just naturally remember that, oh, the opposite would be GEF, and then that would be GTP. All right. So nuclear import, this is a general overview of it. It's a cycle essentially. So that's why there's like this loop. And it starts off with fully folded proteins, as Robert had said in the beginning. So for the nucleus, proteins are localized into the nucleus, fully folded. And so what happens is you have a cargo protein, and it has a nuclear localization sequence or a nucleus localization sequence. It's going to bind to an important. And so this important is what takes it through the nucleus pore. In this case, it's called like the NPC. And once it gets through the NPC, it's now inside the nucleus. So inside the nucleus, you have what we talked about before. At first, RAN is bound to GDP, but then GEF comes in to help it out. The GDP disassociates, so it lets go, and GTP filters in and binds. When important binds to RAN GTP, it causes a conformation here. Can you guys see like where my laser is? Okay, cool. So it causes a conformational shift here, and it lets go of that cargo protein. So now all you have left is it's done its job. The protein is inside the nucleus where it needs to be. But in order for the cycle to continue, you're going to need important to come back out somehow. So that's when RAN GTP takes the important back through the NPC pore. But now it has to somehow let go of the important. And so the way that it's going to do that is through GAP. So this is where the GEF gap comes in handy. So again, GAP causes a gap between the phosphates. So instead of triphosphate, it's going to cleave and hydrolyze off that 3rd phosphate. And now it's going to be a diphosphate, GDP. This GTPase complex is now inactive, so it lets go of the important, and the whole cycle starts again. Are there any questions about that so far?
Don't feel pressured to,
Sorry, don't feel pressured to memorize like every single detail, right? Because on the test, you're going to get this exact diagram essentially on your test. So you can look at it and you can say, oh, I can see the important binds to the cargo protein, which allows it to enter. And then you can see where RAN is GTP, you know, which G is bound to, right? And so don't feel like super pressured. Just
as long as you can like look at this and understand what's happening, you get this diagram. So exactly like Robert said, like don't feel pressure to memorize it. It's more like, if you're given this, can you understand what's happening? And then if Laura gives you a question like, oh, well, if GTP cannot bind in, it's going to constantly stay GDP, Is that going to change anything? Things like that. So if you can look at it and understand what's happening, then it'll better equip you for like if she asks a question about like manipulation and yeah. Oh, did I cut someone off? I think someone might have said something. Will the diagram look the same as it is, or will the labels be taken off? I don't remember. Do you remember?
They're not taken out. Like, I'm pretty sure it is this exact diagram.
Yeah, I don't remember. But either way, I believe Laura's still gonna do like the cheat sheets. You get a like a full like A4 copy paper size paper that you can like write notes on or whatever. And if you wanted to like even just draw out these type of systems, it's always really helpful both for studying and for the exams. But I believe she'll let you know too, like what she provides. So you'll definitely know beforehand whether or not it's labeled. Okay, cool. Okay, well, we'll move on to export then. So nuclear export is similar, but it does have some like caveat differences. So obviously, instead of from being out from the outside in the cytosol to go into the nucleus, you're wanting to take things that are inside the nucleus and bringing it back out into the cytosol. And that's why we say that the nucleus, like the signal sequences, they don't get cleaved off, because you never know when you're going to need a protein or some type of export and import to come in and out of the nucleus. It has to be flexible and fluid. So if you cleave off those signal sequences, it's not going to be able to come in or come back out if needed. So you're starting off with, again, GDB bound. In order to make it active, it's going to be GTP bound. And so once it is GTP bound, this GTPase is active, and exportin can bind to the GTPase and a cargo protein. So something that needs to make it out of the cell, it's going to bind to that exportin. Once it's bound to that exportin, the entire complex makes it out of the cell back through the pore. And again, in order for everything to let go of each other, the GTPase has to become inactive. So it has to become GDP bound again. And then that is done through GAP. So that hydrolyzes the GTP, makes it GDP. Everything lets go. The exportin is free to go back into the nucleus. And then the whole cycle starts again. So I know that's like simplified, but again, like notes are all going to be here, and then the slides are going to be sent out. So you're more than welcome to like look at it as much as you can. But again, it's really about whether or not you're going to be able to understand this picture when given to you. Any questions before we move on? Okay, cool. If you do have questions, like feel free to stop or put in the chat as usual. All right, so the last part of this week's topics are gene expression regulation, or at least like an introduction to the basics of gene expression regulation. So you have, for one, we're going to focus on eukaryotes, and then at the end, essentially, it's the same as what prokaryotes do, but with a little few differences. And those are just shown in like diagrams to help you guys picture it better. So for now, we're going to go through eukaryotes. So for transcriptional regulation, this is the main site of regulation for gene expression. So this is like one of the main ways that proteins regulate proteins are regulated. So you have transcription factors that regulate RNA polymerase by binding to the promoter. Without transcription factors, that's what the little abbreviation is. Sometimes you'll see RNA polymerase cannot bind to the DNA. So for instance, if there was a question and it said that transcription factors are blocked or they're not able to bind, then would you expect transcription to happen? And the answer would be no, because if you don't have transcription factors there, there's nothing for RNA polymerase to help bind to the promoter. And so DNA is not able to transcribe, and you're not even going to have that mRNA sequence at all. And so more specifically, there are regulatory transcription factors. So regulatory transcription factors are gene specific. That means that there's a specific place within the DNA that it can bind to, and it doesn't really have the affinity to bind anywhere else on that gene. You can have positive or negative regulators. So positive regulators enhance transcription. So it means that it's going to help RNA polymerase bind even better. So that means transcription is going to go up and you're going to have even more gene expression. On the other hand, you can have negative regulators, and that just means it suppresses transcription. So it's going to prevent the RNA polymerase from binding. That's going to prevent transcription, and that's going to prevent gene expressions. A little bit more about transcriptional regulation. Chromatins, which are histones wrapped in DNA. So these are the histones and they're wrapped in the DNA. Chromatin packaging determines which genes can be expressed. So you can have two different forms of chromatin packaging. You can have the euchromatin, which is the open state. It's more loosely packaged. So when things are more open, such as this bottom half, you have a lot more space for RNA polymerase to come in and bind, and for transcription as a whole to occur. And so, however, on the other hand, if you have a more closed state, things are more compact together, it's harder for proteins or for RNA polymerase to go in and bind. And so you would have less transcription. All right, so then another form of gene expression regulation is RNA modification and processing. So RNA has to be processed and made stable into mature mRNA in order to leave the nucleus. This is specific, again, to eukaryotes. There are a lot of basically like hardships that RNA goes through in order to leave the nucleus, and if it's not properly protected, it could be very detrimental. So think of it as like of newborn babies going out into the cold. You want to make sure it's like bundled up and protected. Otherwise, the cold is going to kill it. And so how is RNA made stable? There are a few different ways that you guys are probably familiar with. So first is the R5' cap and the poly A tail. So the 5' cap is covalently added to the 5' end of every RNA molecule. And it's essentially prevents the RNA molecule from being chewed away by the nucleus. It does so because the 5' cap, it's, what's it called? It is guanosine rather than like the typical shape of it. You don't need to know too much about it, but essentially it's like a flipped version of what you would normally see. And then that confuses like the nucleases. So then it prevents the front of the RNA from being chewed away. And then you can also have a poly A tail, and that protects the mRNA from being degraded as well. So proteins that bind to the poly A tail are required in order to be exported for nucleus. If the mRNA does not have a poly A tail, it's not going out of the nucleus. The cell won't allow it. But eventually the poly A tail does get chewed away over time. So mRNA, no matter how much protection you put on it, it eventually does get degraded. It's all about longevity and how long the cell wants it to be around. Okay, so then continuing RNA processing, there's RNA splicing that I think is covered in Bio 151 too sometimes. So you have our non-coding regions known as introns within your RNA, and they have to be spliced out before translation because they're not useful to have. And so splicing occurs before leaving the nucleus, because once you leave the nucleus, that's where translation is going to happen. And so again, introns, which are shown as these like empty spaces, they are areas that are not translated, whereas exons are both transcribed and translated. So you want to have a spliceosome, which is an enzyme that comes in to take out these introns, and then it pieces together these exons together. There is something known as alternative splicing, which is also known as exon skipping. And that basically just allows the cells to regulate which exons make it into the final mRNA transcript. So for example, if you wanted only this dark red piece and this last dark red piece, you can take out all of the middle ones, including this exon, through alternative splicing. And this also allows for a lot more diversity within your genome. Okay, so just to show a little bit more about alternative splicing, for example, you could have 1, 2, 3, 4 exons, take out all of the green parts, those were the introns. You could choose to have 1, 2, and 3 together, or you can choose to have 1, 2, and 4 together and take out, when you took out these green introns, instead of cutting 1, 2, 3, 4 times, you just cut 1, 2, and you take that third exon out with it. So that's just alternative splicing. And again, it helps with like more diversity within your genome because you can have a lot more different variations of what you're translating. Okay, so then a couple more different ways of gene expression regulation. So you have to have stability of the mRNA or mRNA degradation. Again, an mRNA lifespan, it's always going to eventually be degraded, but it can definitely be regulated by and determined, like the cell can determine how fast it wants it to be degraded. So that involves RNAi, which is known as RNA interference, but that's something that'll be covered later on in the course, a few weeks later. And so you don't have to worry about that now, but cells can control overall mRNA degradation rates and the degradation of specific mRNAs. So I don't think Laura specifies how, so it's not too important to know how. You just need to know that it is a possibility and that mRNAs do eventually get degraded and chopped up into pieces to be reused, but the cell can determine how fast that occurs. And then, of course, if there's transcriptional control, there is also translational control. So your ribosome binding accessibility is one way to control what gets translated because ribosome is what does the translating. So if you have a protein to block a ribosome from coming, that could stop or prevent or lessen translation. If you have a protein that could help ribosome binding, that will increase translation. There's also, oh, I'm blocking myself. There's also mRNA localization. So this means where the mRNA is located can affect gene expression regulation. So if it's going to a ribosome, it might get taken away from the ribosome. Basically, like you can interfere where the mRNA is going so that maybe before it gets to the ribosome, the cell determines that they don't want to translate the mRNA anymore and then goes to degrade it immediately. So if it takes it away from the ribosome, of course, that means there's no translation. All right, so then this is just a little simplified way to kind of show the difference between eukaryotes and prokaryotes in regards to gene expression regulation. So these are all the ways that we talked about so far for eukaryotes specifically, but it's split up into differences and similarities for both sides. So eukaryotes package DNA into chromatin. You can have euchromatin, which is loose, or heterochromatin, which is tightly packed. However, prokaryotes do not package DNA into chromatin, so they don't have histones. Their DNA is all just loosey-goosey. Eukaryotes also have a nucleus, so obviously mRNAs have to be processed and properly packaged in order to be transported out in order to protect the mRNA, but there are no nuclei in prokaryotes, and so it doesn't need to really process the mRNA or protect the mRNA from anything. It's all just there in the cytosol. And then eukaryotes regulate transcription with transcription factors. Prokaryotes do that as well, but they have different transcription factors. So eukaryotes have basal transcription factors, which are also like known as the regulatory transcription factors. So these are sequence specific. And on the other hand, prokaryotes have sigma factors. More specifically, you'll hear about sigma 70 a lot, but I think that comes up a lot more in further weeks as well. So you'll definitely get familiar with these different transcription factors. And then these are all similarities. So they both regulate how fast mRNA can get degraded. They regulate where mRNA can go if it needs to get taken out early on. They regulate translation with like the ribosomes, and they can regulate protein activity and degradation as well. And then these next two slides are just like a visual interpretation. For me, this helped a lot to see like the order of expression regulation that can occur. So you can look on this on your own time later on, try to get a better grasp of like what's occurring when. And then the next slide just kind of shows the differences. So all the differences really are before the mRNA, in eukaryotes at least, are taken out of the nucleus, because that's the main difference between eukaryotes and prokaryotes. All right, so yeah, that was it for content-wise. Obviously, it's a lot, and maybe you haven't gone to all of it, but hopefully this offers like a baseline kind of to jog your memory for later on when you do get to watching Laura's videos. If anyone has any questions, definitely shout it out now. All right, otherwise I'll stop the recording for now, but we can definitely stay for any questions that people have.